Hi, everyone. My name is Renuka Limgala, and I am a clinical research scientist at a Lysosomal and Rare Disease Research and Treatment Center, which is located in Fairfax, Virginia. Today, I will be talking about immunopathogenesis of lysosomal storage diseases and how they can influence the severity of COVID-19 presentation in these patients. I have no relevant financial and non-financial interest to disclose. So in today's talk, I would be briefly reviewing the role of innate and adaptive immune responses, and as well as the cytokine storm in determining the severity of COVID-19 presentation in patients infected by SARS-CoV-2, and review the immunological alterations and inflammatory responses which are persistent and seen in various lysosomal storage disorders. And then finally, I will appraise the role of these immune abnormalities which are seen in lysosomal storage disease patients and in their response to COVID-19 infections and the disease severity. COVID-19, short for coronavirus disease of 2019, is caused by infection by novel coronavirus SARS-CoV-2. As of today, there are a little more than 900,000 cases worldwide. And if you look at the percentage, 52% have been recovered from these disease, while the rest of them still have the active disease. Out of those, 5%, there is a mortality rate of 5%. And this mortality rate differs from geographical location and country to country. The patients, the COVID-19 patients fall into two types, where some of them have mild to moderate symptoms, which include fever, sore throat, shortness of breath, fatigue, body aches, loss of taste and smell, congestion, and nausea, while a certain fraction of them progress to a severe disease, and the complications include acute respiratory failure, acute respiratory distress syndrome, which is called ARDS, pneumonia, septic shock, multi-organ failure, and recently it has been shown that in some of the children, they manifest the multi-system inflammatory syndrome. If you look at the patients who are infected by this novel coronavirus, about 80% of them are asymptomatic or mildly or moderately symptomatic. And the rest, 20%, they progress to have either pneumonia, and out of them, a small fraction of them go into even more severe complications like ARDS, septic shock, and multiple organ failure. So as soon as the host encounters the novel coronavirus, there is an activation of innate and adaptive immune response, and there is a rapid and coordinated immune response, which is the first line of defense against the virus pathogens. However, in some cases, there is an excessive inflammatory response, and then there is a dysregulated adaptive host immune defense, and this may lead to a harmful tissue damage in the host. And then there is a massive cytokine or a chemokine release, which is termed as cytokine storm, and it has been hypothesized to induce an immune pathology, which results in the acute lung injury and ARDS. So as soon as a patient he is infected by the coronavirus, this novel coronavirus, the disease can be approximately divided into three phases, which is an asymptomatic or mildly symptomatic phase, with or without the detectable phase, as you can see in the above figure. And then in some cases, it progresses to the non-severe symptomatic phase with upper airway involvement. That is where the pneumonia sets in. And in some patients do recover from this stage as well. And then in a small fraction of patients, it progresses to an even more severe, potentially lethal disease with hypoxia, infiltration into lung, and progression to acute respiratory distress syndrome. And at this stage, there is a high viral load. Innate and adaptive immune responses are body's two main lines of defense. When the body encounters the virus for the first time, the first line of defense is the innate immune response, and it starts right after the infection and some of the players are the natural killer cells and macrophages and cytotoxic T cells, NK cells, NKT cells, which lead to killing of the virus and any of the host cells which are damaged by it. The second line of defense is where the macrophages take in the viral particles and then they produce the cytokines which signal to the T and the B cells to produce a long-term defense against the virus. What happens in certain cases is where the macrophages, instead of stopping at producing the cytokines, they keep producing a massive amount of cytokines and chemokines, and which actually recruit the immune cells to the scene. So the inflammatory response that in moderation, it helps the body fight off the virus. But macrophages can also release other signaling molecules called catecholamines that amplify this response further, triggering the release of more and more cytokines. The result is a runaway feedback loop, like a snowball getting bigger and bigger as it barrels down. So this is termed as the cytokine storm, and this is believed to be the major cause of the severe disease in the COVID-19 complications. In severe patients, there are two ways that the immune system can be imbalanced. So in patients with severe complications of COVID-19, there has been a marked decrease in the immune components, like there is a reduced T lymphocytes, both T helper cells and cytotoxic T cells, as well as interferon gamma production, which plays a role in the viral clearance. On the other hand, these patients also show a highly increased amounts of pro-inflammatory cytokines and chemokines. So there are two ways that there is an imbalance in the immune response in the patients with the severe disease. Now, let me briefly take you through the lysosomal storage disorders and give you a bird's eye view about these diseases. Lysosomal storage disorders are a group of about 60 inherited metabolic disorders. They are caused by a deficiency of one of the lysosomal enzyme or its activator or a transporter protein, as a result of which that enzyme it does not function properly. The main role of the lysosomal enzymes is to degrade the substrates. And when these substrates are not degraded properly, they cause a whole bunch of symptoms in the patients suffering from lysosomal storage disorders. So each of them individually is considered a rare disease. However, when you put together these group together, there is an incidence rate of 1 in 8,000 live births. Most of these lysosomal storage disorders are inherited in autosomal recessive manner. However, there are a couple of them which are X-linked as well. Taken together, lysosomal disorder patients show a huge manifestation of pathological symptoms, and they can affect various organ systems. Now, the main focus of today's talk is how the immune system abnormalities are present in several lysosomal storage diseases. There are several lysosomal storage diseases where there is abnormalities in either innate and adaptive immune system have been shown to be persistently present in spite of the treatment. Some of the most common and well-known studied diseases with the immune system abnormalities are Gaucher disease, Fabry disease, Fabry disease, GM1 and GM2 gangliosidosis, both myeloid cell and globoid cell leukodystrophies, Niemann-Pick disease type C, and mucopolysaccharidosis or MPS diseases. Now, coming to some of the immune disorders which are seen in this lysosomal storage diseases, I am going to walk you through some of the findings. Our labs and other labs have shown that there are several T cell and B cell abnormalities in several lysosomal storage diseases. For example, I will start by showing some of the T cell abnormalities that we found in our lab in the patients affected with Fabry disease. As you can see, there is an increase in the CD8 or the cytotoxic T cells when you compare to the normal or the non-Fabry disease subjects. Not just the number of the cytotoxic T cells, we also see, based on the presence of the activation markers, we see that these cytotoxic T cells are chronically activated in the subjects with Fabry disease. Not even that, there is also an elevated number of resident memory T cells as well. And there is a difference between the manifestation of these T cell abnormalities in the pediatric versus adult Fabry disease subjects. Coming to the Gaucher disease, which is probably one of the most well-studied lysosomal storage diseases in respect to the immune abnormalities. Our lab has shown that there is a difference in the CD4 T helper cells as well as CD8 cytotoxic T cells in the Gaucher disease patients, irrespective of whether they're under treatment or not. We also see there is a skewed T helper to cytotoxic T cell ratio, and we see an increase in the memory cytotoxic T cells as well. Gaucher disease is divided into three types, Gaucher disease type 1, and then Gaucher disease type 2 and 3 based on the neurological involvement and severity. In general, GD2 and 3 are supposed to be much severe than the Gaucher disease type 1. And complement system is a part of an innate immune system that enhances the host's defense in clearing the pathogens. C3A and C5A are released when complement system is activated, and these molecules themselves may act as anaphylatoxins and, among other things, may induce a local inflammation. As you can see in the figures above, there is a different amount of C3A and C5A, or the anaphylatoxins, in GD2 and 3, which are more severely affected patients compared to either GD1 or controls. Interestingly, it has been noted that there is a recent publication where when they compared the COVID patients who were in ICU and who are not admitted to ICU, there is a significant increase in the CD14 plus CD16 plus monocytes, which are considered non-classical monocytes in the patients who were admitted to ICU. And they are supposedly they are thought to be responsible for the release of pro-inflammatory cytokines and as a cause of a heightened inflammation in these severe patients. Interestingly, recently we observed that in the Gaucher disease and the Fabry disease patients, there is an increase of this non-classical monocyte subset circulating in these patients. Not just the number of these non-classical monocytes, they are also in a constant level of activation in these patients, both Gaucher disease and Fabry disease patients that we have studied. Recently, there is an idea proposed as a potential positive factor for the cytokine storm when innate immune system does not complete its job of most viral clearance and killing of infected host cells, and adaptive immune response kicks into action. It then slows down the innate immune system, thus delaying the viral clearance. However, when the virus takes root again, the adaptive immune response goes into a hyperdrive and releases more and more chemokines and cytokines in a feedback loop, thus causing the severe disease. So basically, the bottom line is that there is disconnect between the innate immune system and the adaptive immune system, which is causing hyperinflammation and release of pro-inflammatory cytokines, causing the cytokine storm, and thus leading to the severe complications of COVID-19. This slide is more like a review of some of the immune system alterations which are seen in COVID patients with severe complications. And then see if we find a similar or a different kind of findings in the lysosomal storage diseases. Those in the blue in the left column are in the left row are shown that, sorry, in the left column, there is an increase in the pro-inflammatory interleukins and TNF-alpha and the other chemokines like CCL2, CCL3, and CCL4, and there's a macrophage stimulating factor. There's an increase in the non-classical monocytes. There is an increase in the D-dimer, ferritin, troponins in the COVID-19 severe cases. Similar findings have been found in the Gaucher disease patients and Fabry disease patients, and a few other lysosomal storage diseases have also. However, there are certain immune abnormalities which are seen in the COVID-19 cases, but we actually see a reverse of those in the lysosomal storage diseases. For example, interferon gamma, which has a role in the viral clearance, is reduced in the COVID-19 severe cases. However, to begin with, there is an increase or there is an elevated levels of interferon gamma found in the Fabry disease patients. Similarly, there is a decrease in the CD4 T cells or CD8 T cells, which are T helper and cytotoxic T cells and memory T cells in the COVID-19 severe cases. However, there is the CD8 or the cytotoxic T cells or an activated CD4 T cells are seen in elevated numbers in Gaucher and the Fabry disease. Similarly, NK cells, natural killer cells, which are a part of the innate immune system, are shown to be decreased in the COVID-19 severe cases. However, to begin with, in the Gaucher and the Fabry disease cases, there is an elevated numbers for these cell types. Now, based on our findings, let me see if we can come up with a hypothesis as to how COVID-19 would be affecting lysosomal storage disease patients. In the lysosomal storage disease patient, there is already a skewed immune system with respect to some of the persistent immune abnormalities or hyperinflammation. So their steady state itself is a little bit skewed to begin with in the lysosomal storage disease patient. Now, if you add an additional stress to these in the form of, as a result of the COVID-19 infection, there in the scenario one, what happens is there are some pre-existing immune factors which counteract the novel effects of the COVID-19. For example, in the previous slide, we saw that interferon gamma is reduced in the COVID-19 severe cases. However, to begin with, there is an elevated amount of interferon gamma in the Fabry disease patients. So probably it would counteract having a COVID-19 response. And in a similar way, there is the cytotoxic T cells are reduced in the COVID-19 severe cases, but to begin with, they have the Fabry disease and the Gaucher disease patients have an elevated number of these cytotoxic T cells. So probably they would act as a counteracting factor. There is a scenario too, where there is a chronic inflammatory condition in the lysosomal storage disease patients where the body is all the time in an activated state of condition. So in that scenario, if you're presenting an additional stress based on the COVID-19 infection, probably the body is more resilient and it is ready to act on that kind of an external stress compared to the non-lysosomal storage disease patients or the compared to controls. Or in the third case, where the additional COVID-19 related stress actually worsens the chronic inflammation and the chronic immune abnormalities, which are already present in the lysosomal storage disease patients. So these are the three scenarios or the possible scenarios that can happen in a lysosomal storage disease patient when they are affected by or infected by this novel coronavirus. So to summarize my talk, I would like to emphasize that there are a subset of COVID-19 patients who suffer from severe complications and as a result of an abnormal immune response, which is termed as the cytokine storm. And there is an interplay between the innate and adaptive immune responses, which may be the cause for this cytokine storm and the resulting severity of the COVID-19 disease. Now, some of the immune abnormalities, which are seen in a persistent way, and the increased inflammation seen in these in the lysosomal storage disease patients, are also chronically are observed in these severe COVID-19 patients as well. So I will end with there are the two possible scenarios as to what will happen if the lysosomal storage disease patients are infected by this novel coronavirus. Would the chronic immune abnormalities and an increased inflammatory condition make them more susceptible to the additional stress of the COVID-19, or would it make them more protected against the severe complications of COVID-19? Again, it would not affect the rate of infection of the lysosomal storage disease patients. However, we are talking about the protection against the severe complications of the COVID-19. Thank you for listening. If you have any questions, you can reach out to me after at the end of the session, or you can email me.